So hi everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry, and in today's session we will be seeing about various kinds of applications of your molecular technology. And the first and the foremost what we are going to see today is about recombinant DNA technology. Clear? So before going into the proper session, I just wanted you all to go through this MCQ first. Okay. So, which is true about recombinant DNA technology? It is an in vitro cloning process. It is used to multiply all segments of DNA. It requires a primer and the vector is used for DNA transfer. So, what do you think is the right answer for recombinant DNA technology? The right answer is the vector is used for DNA transfer. Okay, so why this is the right answer? If you want to find out the answer, I think you will be easily finding out the answer once we complete this session. So with this introduction, I will just move on into our proper recombinant DNA technology. So what is it actually? What is recombinant DNA technology? It is nothing but a cloning process. So what is cloning process? Cloning process is producing identical copies. From a parent copy, you are producing an identical copy. That is called as a cloning process. So in this case, this cloning process is done in vivo, in a cell. But we are the one who have manipulated that cell in such a way that they clone only our copies which we are interested in. Okay. So finally, what is the result of that? We get a clone of the desired DNA fragment. Okay. So this is all about your recombinant DNA technology. It is nothing but the process of replication. It is simply the process of replication wherein from the parent strand you will be getting two daughter strands. Okay. But this replication is not occurring naturally. You have made some kind of a manipulation in that genome and you wanted to manipulate and you wanted to amplify or clone that desired DNA fragment which is of your interest. Okay. So this is carried out in a cell based manner. In a bacterial cell, for example E. coli, if this is carried out in a cell based manner, this technique is called as recombinant DNA technology. If instead of a cell based cloning, if this is a cloning which is being carried out in vitro, that is inside a lab, inside some kind of an instrument called as thermocycler, this is called as polymerase chain reaction, otherwise called as PCR. So are you clear about the difference, guys? PCR is in vitro amplification. Okay, it is in vitro cloning process. This is nothing but PCR, polymerase chain reaction. It is in vitro cloning process. Whereas what recombinant DNA technology is about in vivo cloning process. It is a cell based cloning process. So it is used to multiply all the segments of DNA. You should be knowing the answer by now. It is not all the segments of DNA. It is the specific. It is not all. It is specific segments of DNA which we are interested in. There is no point in multiplying all the segments of DNA because what we are doing in recombinant DNA technology is that you are incorporating a fraction, a part of our eukaryotic genome into a prokaryotic gene, okay, into a prokaryotic DNA, and after that you are trying to multiply that. Okay. So there is no point in multiplying all the segments of eukaryotic DNA in a prokaryote. There is no point in that. Okay. So you are multiplying only the specific segments of DNA. Okay. Primer is needed. This primer will be required for polymerase chain reaction in order for the DNA polymerase to occur, to function. In case of recombinant DNA technology, that problem will be taken care by the E. coli bacterium itself, where the thing will get divided easily. So, primer actually is not needed in recombinant DNA technology, and instead of primer we use a vector, okay, and that vector will be used for transfer of our desired DNA of interest. That is why this is the only true answer about recombinant DNA technology. Are you clear, guys? Fine. So, with this we move on into the steps of recombinant DNA technology. So, what are these different steps involved in this recombinant DNA technology? First, you have got an entire genomic sequence, okay, of which this one which is highlighted in green will be your gene of interest.